Welcome to part 1 of the plant diversity video, bacteria presented by Kalia on behalf of Achyut Mallik.
This section of the video focuses specifically on bacteria. In the introduction to bacteria, we learn the term was coined by Ehrenberg and discovered by Leeuwenhoek. Bacteria belong to the kingdom Monera. They are unicellular, prokaryotic, and microscopic. The study of bacteria is called bacteriology, and the father of bacteriology is Robert Koch. Bacteria are ubiquitous, found everywhere except distilled water. Their nucleic acid includes RNA plus DNA, and a commonly studied bacterium is Escherichia coli. Louis Pasteur is credited with giving the term virus, and he is also considered the father of microbiology. His contributions include discovering the rabies vaccine and developing techniques like sterilization and pasteurization. He is also associated with the theory of biogenesis. The average size of bacteria ranges from 2 to 10 micrometers. The smallest bacterium is Dialister pneumocyntes. The largest bacterium is Beggiatoa mirabilis, and the longest bacterium is Bacillus buccilli. Bacteria are classified based on several criteria. There are 5 types based on shape, 6 types based on flagellation, 4 types based on their role in the nitrogen cycle, and 2 types each based on their capsule and cell wall. There are several shapes of bacteria, including rod-shaped or bacillus such as lactobacillus. Spiral-shaped are spirillum, for example, Treponema pallidum. Spherical are coccus, such as Streptococcus. Comma or C-shaped are Vibrio, like Vibrio cholera. Irregular or not fixed shapes are pleomorphic, such as Azotobacter. This slide visually illustrates the different shapes of bacteria: coccus, bacillus, spirillum, Vibrio, and pleomorphic. The types of flagella in bacteria include Atricus, found in Lactobacillus and Pasteurella; Monotrichus, such as Vibrio cholera; Cephalotrichus, seen in Spirochaetae and Treponema; Amphitrechus, like Nitrosomonas; Lophotrichus, for example, Pseudomonas, and Azotobacter. Finally, Paratrichus, including E. coli, Salmonella, and Shigella. Here are visual examples of bacteria with different types of flagella: Atrichus, Monotrichus, Amphitrechus, Cephalotrichus, Paratrichus, and Lophotrichus. The nitrogen cycle involves several steps with distinct features and bacterial examples. Nitrogen fixation converts atmospheric nitrogen to ammonia compounds using rhizobium. Nitrification converts ammonia to nitrite using Nitrobacter. Ammonification converts protein to ammonia using Bacillus vulgaris. Denitrification converts nitrates back to nitrogen gas using Pseudomonas. This diagram shows the cell structure of bacteria, illustrating key components such as the capsule, pilus, cell wall, plasma membrane, cytoplasm, ribosomes, nucleoid, and flagellum. The bacterial cell structure is divided into wall layers and matrix components. Wall layers include the capsule or slime layer, cell wall, cell membrane, flagella, and fimbriae or pili. The matrix includes the cytoplasm, nucleoid or genophore, and episomes. The capsule is the outermost layer of the bacterial cell, chemically composed of polysaccharides and amino acids. Based on the presence of a capsule, there are 2 types of bacteria: R strain, which lacks a capsule and is non-virulent, and S strain, which has a capsule and is virulent and pathogenic. The S strain is deadly because it produces a protective slime coating that helps it evade the mouse immune system, causing pneumonia. The R strain is harmless. It does not produce a protective slime coating and is easily defeated by the mouse immune system. The presence of a capsule in any bacteria increases its virulence or ability to cause disease. It acts as a barrier to the immune system, preventing phagocytosis because the encapsulated bacteria are barely exposed. Examples of encapsulated bacteria include Escherichia coli, Pseudomonas aeruginosa, Klebsiella pneumoniae, Haemophilus influenzae, Streptococcus pneumoniae, and Salmonella typhi. The cell wall is the outermost layer of the bacterial cell, chemically composed of peptidoglycan, muramic acid, murane, or mucopeptide. Based on the cell wall, there are 2 types of bacteria: gram-positive and gram-negative. Gram-positive bacteria have a smooth cell wall with an outer membrane present. Gram-negative bacteria have a wavy cell wall with the outer membrane absent. The comparison between gram-positive and gram-negative bacteria continues. Gram-positive bacteria stain purple, violet, or blue, while gram-negative bacteria are colorless or red. The cell wall is simple and thick for gram-positive but complex and thin for gram-negative. Teichoic acid is present in gram-positive but absent in gram-negative. Flagella and fimbriae are absent in gram-positive but present in gram-negative. Pathogenicity is less in gram-positive bacteria and more in gram-negative bacteria. Gram staining technique begins with bacteria staining all bacterial cells a purple, blue, or violet color. The stain is then fixed in the bacterial cell. This image visually represents the process of gram staining, highlighting the differences between gram-positive and gram-negative bacteria during the procedure. The cell membrane, also known as the plasma membrane, is chemically composed of lipids and proteins, specifically phospholipid. It lacks sterols. The enfolding structure of the cell membrane is the mesosome. In photosynthetic bacteria, the cell membrane is the thylakoid. Flagella are composed of flagellin protein and are an extension of the cell membrane. They arise from the blepharoplast of the cytoplasm. The 9+2 arrangement is absent in bacterial flagella, unlike in eukaryotic cells where it is made of tubulin protein. Fimbriae, or pili, are composed of pillin protein and are commonly found in gram-negative bacteria. Pili can be of two types. The cytoplasm of bacteria contains a matrix that includes ribosomes and cytoplasmic inclusions. Examples of cytoplasmic inclusions are glycogen, poly-beta-hydroxybutane granules, sulfur granules, and gas vesicles. Regarding genetic materials, a true nucleus and nuclear membrane are absent. The DNA is a double-stranded circular structure. This DNA is naked, meaning it is without histone protein, and is freely scattered in the cytoplasm. Extra genetic materials include plasmids, such as R plasmid, which provides resistance to antibiotics, NIF plasmid, involved in nitrogen fixation, and F plasmid, which is a fertility factor. Episomes are also considered extra genetic material. Nutrition in bacteria is broadly divided into autotrophic and heterotrophic nutrition. Autotrophic nutrition includes photoautotrophs and chemoautotrophs. Heterotrophic nutrition includes saprophytic, parasitic, and symbiotic types. Autotrophic nutrition includes photoautotrophs, which use photosynthetic pigments like bacterial chlorophyll and sunlight to form food. Only photosystem I is involved, except in cyanobacteria, where both I and II are involved. Chemoautotrophs lack photosynthetic pigments and form food by chemosynthesis, using the oxidation of inorganic substances as an energy source. Examples include iron, sulfur, nitrifying, hydrogen, and methane bacteria. Heterotrophic bacteria are classified into three types: saprophytic, which obtain food from dead organic matter; parasitic, which obtain food from the host body; and symbiotic, which live in a mutual relationship where both the organism and host benefit, such as Rhizobium and E. coli. Respiration in bacteria is classified into obligate aerobes, which live only in the presence of oxygen, like Azotobacter and Bacillus, and obligate anaerobes, which live only in the absence of oxygen, such as Clostridium. The third type is facultative aerobes. They usually live in the absence of oxygen but can live in the presence of oxygen as well, for example, Chlorobium. The fourth type is facultative anaerobes. They live in the presence of oxygen but can also survive in its absence. Next, let's explore reproduction in bacteria. The three main methods are vegetative, asexual, and genetic recombination. Vegetative reproduction, most common in coccus, involves binary fission and takes about 20 minutes, with the number of bacteria becoming 2 to the power of n. Asexual reproduction involves endospore formation. This process is thin-walled and highly refractile, representing reproduction without multiplication. The third method is genetic recombination. Genetic recombination is an important part of bacterial reproduction. The three types of genetic recombination are bacterial conjugation, transformation, and transduction. Each of these methods has unique features and was discovered by different scientists, which we will explore further. This image illustrates bacterial conjugation, a process of genetic material transfer between bacterial cells through direct contact. Here we see a visual representation of the transformation process in bacteria, where genetic material is taken up from the surroundings. This diagram depicts the process of bacterial transduction, which involves the transfer of bacterial DNA by a bacteriophage. This slide provides another illustration of bacterial transduction, showing the bacteriophage facilitating gene transfer. Now let's look at the economic importance of bacteria. One major role is in antibiotic synthesis. Bacteria are also responsible for various diseases. For example, typhoid is caused by Salmonella typhi. Tuberculosis is caused by Mycobacterium tuberculosis. Pneumonia is caused by Streptococcus pneumoniae. Whooping cough is caused by Bordetella pertussis. Other diseases include tetanus, diphtheria, gonorrhea, meningitis, syphilis, leprosy, cholera, plague, bacillary dysentery, and gastroenteritis. We finish with some special points. The pigment present in cyanobacteria is C-phycocyanin. The correct sequence of phases in the bacterial growth curve is lag, log, stationary, and decline. Sexual reproduction is absent in cyanobacteria. The sex factor in bacteria is F. replicon. Black death is plague, caused by Yersinia pestis. Bacteria possess one chromosome, and the most suitable pH for their growth is 7. Moving to mycoplasma, E. Nocard and E. R. Raux discovered these organisms in 1898. Mycoplasma, or PPLO, are the smallest, simplest, unicellular, non-modal, free-living, and highly pleomorphic organisms. They lack a cell wall, leading to the name, quote, molecule-like organism. They multiply by binary fission and elementary bodies. They cause diseases in plants like little leaf of brinjal and clover dwarf, and in humans, such as mycoplasmal urethritis and pleuropneumonia. This image provides a visual representation of mycoplasma's structure. Thank you for watching this video on bacteria. Here is a quick MCQ question for you. Q. Nitrosomonas is A. Phototrophic, B. Symbiotic, C. Chemotrophic, D. Photosynthetic. Q. The bacteria found in the root nodules of Leguminaceae is A. Rhizobium, B. Thiobacillus, C. Bacillus, D. Clostridium. You can reach out to BAM Sir via email.